Okay, so we are going to be discussing the development of the hindgut. And in relation to that, we are talking about the partitioning of the cloaca. This is an embryological process that takes place in the 5th to 7th weeks of intrauterine life. And I'm going to draw the successive stages which are involved in the partitioning of the cloaca. So in the 5th week of intrauterine life, as we can see, I'm going to draw a sagittal section through the embryo, and that is going to represent the lower part of the embryo. And if you recall from the previous lectures, that there are 3 parts of the primitive gut, that is the foregut, midgut, and then the hindgut. So here we can see is the midgut, forming the primary intestinal loop, which enters inside the umbilical cord. And then we have the hindgut. This hindgut will enter a pouch-like diverticulum at the lower part or the caudal end of the embryo. This pouch-like diverticulum is known as the cloaca. So the hindgut, it enters the posterior end of this pouch-like region in the caudal part of the embryo known as the cloaca, whereas the anterior part of this cloaca receives the allantois. The allantois is the diverticulum which enters the umbilical cord, and at one side and at the other side you can see it is entering this here pouch-like diverticulum, that is the cloaca. So why don't we label these structures? This here was the midgut. Just going to write MG for now. And the lower part is the hindgut. The hindgut is entering this pouch-like region, which is the cloaca. Okay. And then you can see this is the allantois, which is entering the anterior side of the cloaca. In the caudal region of the embryo, the cloaca is in communication with this pouch, which is the cavity right now. This is the external area, which is known as the proctodium. So the proctodium, is lined by the ectoderm, whereas this whole region, which is yellow, that is the region which is lined by endoderm. Proctodium is lined by ectoderm. So the junction point of the ectoderm with the endoderm, this here is known as the cloacal membrane, where ectoderm comes in direct apposition with the endoderm and there is no intervening mesoderm. So in the 5th week of development, this is the cloaca and it is receiving the allantois as well as the hindgut, right? We know that the hindgut is going to form eventually the anorectal canal, okay? And we also know that the allantois over here will eventually form an embryological derivative known as the urogenital sinus. So allantois will form urogenital sinus, whereas the hindgut will form the anorectal canal. Now, why does this process occur? Let's look at the mesodermal tissue which is present between this mesodermal tissue, which is present between the allantois and the hindgut. This wedge of mesoderm is known as the urorectal septum. So I'm going to label it. This is the urorectal septum. Now what happens is that this urorectal septum grows caudally, pushing the allantois and the hindgut further apart. Now let's move on to the next further stage of development in the next coming weeks. So we said that the hindgut and the allantois, they are going to form these embryological derivatives, which are anorectal canal and the urogenital sinus respectively. So let's again draw this cross section through the lower half or the caudal part of the embryo. Okay, so the allantois, which was initially a narrow structure, now its upper part becomes dilated and the lower half is only narrow and it is entering the cloaca. Whereas when we look at the hindgut, it has also dilated and it is entering the posterior part of this cloaca. So now we can label the hindgut and we can also label this here structure, which is now the urogenital sinus. Hindgut is now called anorectal canal, okay? Because it's going to form the rectum and the superior part of the anal canal. And whereas this allantois has now dilated and formed the urogenital sinus. The urogenital sinus will eventually form the urinary bladder and the urethra. So its upper part is dilated to form urinary bladder and the lower part is still narrow and it forms the urethra. Both of these structures are entering the cloaca, but as you can see that urorectal septum, which was present between these two embryological regions, this has further moved caudally closer to the cloacal membrane. The cloacal membrane will eventually rupture, and if you recall from your previous knowledge, it's going to rupture in the 7th week of intrauterine life. So let's just move on, get a fresh page over here and look at the further stages of development. In the further stages of development, once again, I'm drawing for you that caudal region of the embryo. And now we can see that as the cloacal membrane, it ruptures in the 7th week of development. This establishes separate openings for the urethra. This is the anterior opening, which will be the opening of the urethra, whereas the rectum and the upper part of the anal canal, that now has a separate opening, which is posterior to the urethral opening, known as the anal opening. So what about the urorectal mesoderm, the mesodermal tissue that was present between these endoderm line cavities? This urorectal mesoderm has eventually reached the lower edge of the ectodermal region, and at that point it forms a structure which is known as the perineal body. This here is the perineal body. It is a supporting structure present in the perineum, and it is placed between the urethral opening anteriorly and the anal opening posteriorly. So I'm going to label this as the anal opening, and anteriorly this here is the urethral opening, okay? So this is the partitioning of the cloaca. You can see the cloaca has now subdivided into these two separate regions. One is the region for the bladder and the urethra, and posterior to that is the anorectal region. And these are the places which are lined by endoderm. And this is the area which was the proctodium. Proctodium was lined by ectoderm. And it forms the lower 1/3 of the anal canal. And these are the regions of the anal canal which have a dual embryological origin. So superior 2/3 of anal canal is derived from endoderm, and the lower 1/3 of anal canal is derived from the ectoderm. So this these were the successive stages which were involved in the partitioning of the cloaca. Okay, thank you. So we're done with that.